So,
The film, could you try to...
Very much. Good afternoon, ladies and gentlemen, dear colleagues. Thank you very much for giving me the opportunity to share with you some information and data about COVID-19 testing and SARS-CoV-2 in the context of the pandemic. These are my disclosures. I just want to mention that the data that I present with you, of course, is as per very recent, and the field is moving very quickly in SARS-CoV-2. In this presentation, I will address the COVID-19 disease and SARS-CoV-2 diagnostics. We will discuss disease management, and I will very briefly touch on upcoming vaccinations. Diagnostic tests are an essential service in the delivery of healthcare. In vitro diagnostic testing has become an indispensable tool in clinical practice. It can provide critical information at every step of the patient pathway, and results in vitro testing influence as many as 70% of the clinical decisions. Never has this been more true, as the COVID-19 pandemic has opened a whole new window into the world of diagnostic testing. The COVID-19 pandemic has disrupted the life of billions of people, nearly overwhelming healthcare systems and pushing the global economy to the brink of collapse. So the quest for effective treatments and a preventive vaccine starts with and depends on the broad availability of fast, accurate, and reliable testing. In vitro diagnostic testing plays a vital role in testing who may have contracted COVID-19, ensuring that healthcare professionals can work safely and that quality care and treatment is available to all those who need it. Helping to flatten the curve, lowering the rate and spread of infection by providing governments and healthcare institutions with actionable test results. High-quality diagnostics aid more accurate disease identification and leading to more appropriate patient care. So what are coronaviruses? They are relatively large viruses compared to other viruses we know. They have a diameter of up to 120 nanometers. They are enveloped RNA viruses with a single-stranded RNA genome, and the RNA is largely covered with a protein called the nucleocapsid protein. The nucleocapsid protein is one of the 4 structural proteins of the coronavirus. The others are the membrane protein, the envelope protein, and the spike protein. And we will hear about those 3 other proteins during the rest of this presentation. Coronaviruses, as most of you know, are zoonotic, which means they are transmitted between animal and people, and the animal reservoir are ecologically diverse with the widest variety seen in bats, which is also the reservoir for many of the viruses. Mammals may serve as intermediate host, facilitating recombination and mutation events with an expansion of the genomic diversity. There are 7 known human coronaviruses with varying harm factors. 4 endemic common cold viruses responsible for 5 to 10% of all upper mild and lower respiratory tract infections, and 3 severe respiratory syndrome viruses with high fatality rates. So these are SARS-CoV, SARS-CoV-2, and MERS-CoV. 2 previous outbreaks have been observed. One is the SARS-CoV in 2022, and the other one is the MERS-CoV in 2012. SARS-CoV-2 is the cause of COVID-19 disease. The transmission is from person to person via respiratory secretion and contaminated surfaces. Incubation time 2 to 14 days. Main symptoms are fever, respiratory symptoms, anosmia, and it's worth to say that probably anosmia is the main characteristic symptom that we observe at the moment. If you have a patient that comes in with anosmia and a combination of any of the other symptoms, you can be quite certain that the test will be positive. However, clinical presentation ranges from asymptomatic to mild illness, pneumonia, or even fatal disease. A number of patients do progress into very severe cases with case mortality rates ranging between 0.8 and 15%, depending on the country, of course. The risk factors for severe disease and death are old age, male sex, comorbidities, obesity, and smoke. And at least in Switzerland, obesity is probably the highest and the greatest risk factor that we see. The patients that are currently hospitalized in intensive care have a number of them are very obese. So that's one of the risk factors that we in Switzerland see for severe disease. After the virus enters the body and reproduces, an immune response is triggered and antibodies are formed. During this period, the patient is deemed infectious and can pass on the infection. Once the virus is eliminated by neutralizing antibodies or cellular immune response, the patient recovers. Again, here it is important to note that even if small amounts of virus are present, along with antibody response, the patient may still be infectious. With each phase of the infection, typically clinical symptoms occur, and the clinical symptoms can be evaluated using a range of diagnostic throughout each phase as the infection progresses. If we look at the disease progression, there are two main phases. The first one is called the viral response phase. In this phase, the virus itself causes the disease symptoms by infecting cells and lysing those cells during proliferation. This is then moved over into the host inflammatory response phase, which is characterized by the body responding to the infection. And in roughly 5% of the cases, it can lead to hyperinflammation. This is in the end what causes the severe symptoms and can eventually lead to death. Now let's look into the types of markers and the development throughout the course of the infection. In ongoing infection stages, viral RNA and antigen appear usually quite quickly and have a steady decline to convalescence, and measured viral ANA and antigen are indicative of an infection and, of course, infectivity. Once the immune response begins and antibodies to the virus start to develop, the levels of RNA and antigen drop significantly. The first antibodies to appear are IgM, closely followed by IgG and neutralizing IgG antibodies, and seroconversion takes place around the medium day of 12. The infection is then said to be resolved and potential immunity is gained. However, more studies are required to understand the longer-term antibody profile and immunity levels of the patients. In SARS-CoV-2, the antibody pattern differs a bit from textbook serology dynamics. We see increasingly simultaneous rays of IgM and IgG. The two primary important questions when using IVD testing for COVID-19 are: first, is the patient shedding virus and is potentially infectious to other people? And the second one, has the patient recovered from a previous infection and developed antibody response to the virus? It should be noted that currently there is no evidence that people who have recovered from COVID-19 and have antibodies are protected from a second infection. However, the cases that are described in the literature are fairly anecdotal, and from the confirmed cases, for example, the Hong Kong patient, it's known that the new infection with SARS-CoV-2 had a slightly different genomic profile. For the time being, there is no robust evidence that patients get reinfected with exactly the same strain again. So when to use which tests? And again, there are the two main questions you ask yourself as a clinician. Is somebody actively infected? You want to look for tests that detect the virus. Those are either RT polymerase chain reactions, and these tests are available in central lab testing or in point of care testing. The second methodology is looking for protein, virus protein in antigen testing, and there as well there is central lab testing available as well as point of care testing. And the second question is, has somebody been previously exposed to virus, and is there potential immunity? And that is accomplished with antibody testing, here again, either available for high throughput and automated central lab testing, or more ad hoc in a point of care setting. If we take a look at the individual tests and the intended uses, the SARS-CoV-2 PCR test is probably the gold standard for testing of symptomatic patients. There is one caveat with testing for PCR, is that these tests are so sensitive that they will pick up signal of even virus particles that are no active virus anymore. To kind of avoid that and to really filter out potentially infectious people and particularly super spreaders, there is also antigen testing available, which is less sensitive and less specific than PCR. However, it will definitely pick up super spreaders and people that have a very high viral load. So the use case here as well is you have symptomatic patients or asymptomatic patients that have a proven exposure to risk patients where you can test if somebody needs further attention or isolation, etc. The antibody test for seroepidemiological surveillance is anti-SARS-CoV-2 nucleocapsid. That is a qualitative test. I mentioned already earlier that is a test in which you want to find out if a patient already had exposure to SARS-CoV-2 and had gained potential immunity. There is another antibody test which is very important to understand. That is the anti-SARS-CoV-2 S test, and S stands for spike protein. That is a quantitative test, not a qualitative test as the N test. And this test, as it looks for spike protein, can be used to reliably determine immune response within a vaccination setting, because the vaccine candidates that are currently under development will, most of them, target the spike protein as a vaccine target. So to understand the quantitative amount of S antibody is helpful in either vaccine settings as well as in serum samples for convalescent plasma therapy, where you want to understand does a patient carry a certain amount of potential antibodies. Quick look at the patient journey. Basically, there are subclinical prodromal phases, the period of illness, the critical illness, and then, of course, the recovery. The patient comes in and gets tested with PCR or antigen testing, and when you have a positive test result, you do the clinical follow-up and the isolation with COVID-19. In case of a negative test result plus continuation of symptoms, you may want to resample or retest. There again, PCR testing is done. You may want to look at other respiratory infections as well, tuberculosis, something which is still very, very prevalent, and of course influenza A and B testing. At the moment, we also have tests available that do test both for influenza A and B plus SARS-CoV-2, and that is particularly helpful for seasonal flu infections where you want to differentiate between influenza or SARS-CoV-2. When we look at the period of illness, you have a patient that has been diagnosed with COVID-19, and either you go into a mild course or you progress into severe course. At the moment, still home isolation for mild courses. If patients go into convalescence, that is, likely to trigger immunity, and the test that you do with patients that are COVID-19 positive and that have COVID-19 disease is listed here. So it's not only that you look for SARS-CoV-2, but you also kind of monitor patients with regards to other parameters. For what we know now about COVID disease is that it has a quite a dominant impact on cardiovascular system. So yeah, that may be something interesting as a workup as well. When looking at the period of critical illness, you have ICU submission, mostly triggered by lung deterioration, which the ventilation of the lung is fairly okay, but you don't have oxygenation. So it's interesting that you don't see the same amount as a fluent pneumonia. You have quite dry pneumonia, but the oxygen transmission still is not sufficient for these individuals. And the clinical or the lab workup here is listed as well. So you kind of monitor patients for further progression of disease, for potential sepsis, etc. And finally, the surveillance. Again, patients that are mild and go into convalescence should go home in isolation, and remote follow-up may be applicable. And in the later run-up, you still want to trigger and follow up with some of the cardiovascular parameters. Yeah, quickly, this slide is what's the critical importance and what features must an assay have. And I think I want to quickly touch on both the tests that I introduced earlier. The antigen assays and the PCR needs to have a very high sensitivity. This means they must be very reliable, detect the presence of virus, and that is, since false negative, depict missed cases that can lead to high risk for an infected patient to further spread the virus. With antibody testing, it's a bit different because antibody needs to have a very high specificity. It means that they must be very precise in telling that a result is truly positive, since false positive results lead to the erroneous assumption of convalescence and putative immunity, and that is putting the person at risk of infection by otherwise preventable exposure. I want to show this at a very quick example when we discuss specificity and positive test result. Prevalence is very important. So due to prevalence, a positive test result in New York, which is considered a high prevalence location for SARS-CoV-2, may be very different from a positive test result in Santa Clara in California, where the data suggesting a low prevalence. The same can be extrapolated to distinct populations with different exposure. For example, an infectious disease healthcare worker compared to a blood donor who respected confinement measures and had little exposure to the virus. So the performance parameter we would look at in this case is the positive predictive value and the negative predictive value. I don't want to educate you on sensitivity, specificity, and positive and negative predictive value because I'm certain that yeah, everybody from the audience is aware. I just want to make the case that when we speak about COVID-19 testing, it's important to also look at prevalence. And I'm saying this because there are as well discussions ongoing with regards to pooling. Pooling makes sense in a low prevalence setting. Pooling probably is much more labor intensive and prone to error when you have a higher prevalence within societies. And I guess with the global pandemic at this point in time, we already have moved to a prevalence where pooling, to justify pooling, the extra efforts, the potential errors, etc., must be critically questioned. So quickly, let's take a look at the vaccine candidates. There are different types of vaccines that are in the pipeline. The first one is viral vector vaccines. Those are vaccines that use virus to deliver coronavirus genes cells, and the cells make viral protein provoking an immune response, but the virus cannot replicate. There are whole virus vaccines that use a weakened or inactivated version of the coronavirus to provoke an immune response. There are genetic vaccines. Those use one or more of the coronavirus genes. And there are protein-based vaccines that are usually vaccines that use coronavirus protein or a protein fragment to provoke an immune response. This is my last slide. These are the vaccine candidates in phase 2 or in limited use. All of them include spike protein in some form. They're either part of a whole virus vector or as an individual antigen, and in most of the studies testing for antibodies and or specifically anti-receptor binding domain antibodies is part of the study protocol. So I'm very confident that there is exciting data coming up in the coming weeks and months. Thank you very much for your attention.
Thank you, Dr. Hem. Thank you for this scientific talk. We can move directly to our the booth. Will move to Biorad now. Our fourth speaker, Mr. Nico Fintbol. Mr. Nico is a scientific and professional affairs manager for Quality System Division in Biorad Laboratory Quality System, and he is member of CLSI QMS 24. He will give us a talk, a 20-minute talk about evaluation, mean and standard deviation, and how to perform QC cross-cover studies according to CLSI C24. Mr. Nico, the booth is yours. Mr. Nick. Yes, Mr. Nick, can you hear us? Mr. Nick, please. شباب الصوت فيه مشكلة ولا؟
Hello, hear me?
Yeah, hi, Mr. Nick.
I am still waiting for control of the slides.
عندي تجتمع.
Hello, Mohammed?
Doctor? Please, from all the doctors, can you start your webcams and the mics? Because now it's discussions time. Dr. Cloud, Dr. Wim, Dr. Jim. Yes. Dr. Cloud, can you try to open your mic and the camera, please? دكتور علي صوتك مو واضح. دكتور علي, we can't hear you. Okay. I think the only one he can try his mic, Dr. He's not can open his mic, Dr. Jim, but he can hear us. Dr. Jim, can you hear us? Okay, perfect. دكتور علي, بس صوتك مو واضح. لا دكتور علي, المايك عندك مقفل, لو تجرب تقفل المايك وتشغله.
ايوه سامعني كده؟
ممتاز, سامعك الحين.
في محاضرة باقية لسه, في دكتور آخر محاضرة, دكتور نيك.
دكتور نيك.
Mr. Nick, are you here? Are you here us?
Yes.
Yes, you're talking now.
I can hear you.
Okay.
You can start your talk now directly, please.
Okay, and could they give me control for the slides? Okay, it's coming.
Please, Dr. Nico?
Yes?
Yes, I'm going to upload your presentation now, okay?
Okay,
Yeah, it's here.
now it's coming.
Yes. Okay, sorry for this missing times. Now we can start, Doctor.
Okay, well, welcome everybody. So I want to talk to you today about evaluation mean and standard deviations and how to perform QC crossover studies according to the latest CLSI C24 protocols. So for our objectives, we're going to take a look at explaining how there's two different methods to set up and monitor a mean and a standard deviation. We will also look at using a Levi-Jennings chart to do the interpretation of our evaluation mean and SD. And then as last objective for this morning, we're also going to describe the new simplified CLSI guidance to perform crossover studies in the lab. So when we talk about an evaluation mean and SD, this is something which is very important because every time we talk about quality control, we talk about Westgard rules, we talk about detection and rejection of results, but everything is based on what we are going to use for our evaluation mean and SD. Our target and our standard deviation are the most important settings because we can have the best system if our settings are too wide or too narrow, we will still have problems detecting specific errors. So if we look at the definitions of our mean and SD just as information, I think most people will already know these, but we use the mean, the sum of all the values divided by the number of the values, as our target or our center line. And then we use the standard deviation as our range. So the standard deviation is the sum of all the differences between the values and the mean, and that is then normalized as a value to evaluate. But what also is really important and is used a lot in the labs, because standard deviation is expressed as an absolute value, it's a lot easier for us to use the coefficient of variation. So that is just expressing the standard deviation as a percentage. And we will see that for specific QC events, when we move from one QC control, one QC lot to another QC lot, it can be very important to see and understand the difference between the standard deviation and the coefficient of variation. When we are setting our evaluation mean and SD, there's two major ways that we can follow up our quality control. Either we can set floating statistics or we can fix our statistics. As I've traveled around, I think most of the labs in the world use fixed statistics now, but originally when Dr. Westgard started with all his QC theories, he talked about floating means most of the time. And the difference is that with a floating mean, every time you add more data points, they are added and they update the mean and the standard deviation. When you use a fixed mean and SD, then you will set these mostly after your evaluation period, and then they don't change anymore. So even if results are added, they stay the same. And you can see there that both have some different ways of reacting. So for fixed mean and SDs, because your values don't change, people also feel that they have a feeling of control because they know today, tomorrow, the next day, the mean and the standard deviation will be the same. When you work with a floating mean and SD, then especially at startup of a new lot, for example, your mean and your standard deviation can move a little bit. The more data you get, the more a floating mean almost becomes a fixed mean. If you have 100 data points already with a value of 100, then it takes you another 100 data points with a value of 105, for example, before your mean will move to 102.5. So that is very stable once you get more data. And for that floating mean and SD, I've heard a lot of labs that don't like to use this because they worry that the mean might float away slowly and drift up, for example, and they wouldn't notice. But again, if you have quite some data, even a floating mean is very stable. So how do we look at these means and SDs? Well, everybody will know that we use the Levi-Jennings chart. And in the Levi-Jennings chart is just a visual representation of our QC data points. And the typical part for a Levi-Jennings is that in the middle you have your mean value, and then you have your standard deviations expressed, positive standard deviations above the mean line, and then negative standard deviations below. This also helps us to identify really quickly if there are specific trends or shifts that are going on, and if there are specific events we need to monitor. Typically, Levi-Jennings charts will have limits of 3 standard deviations on the top and -3 on the bottom, but sometimes you might even open them up a bit further to see more variation. But when we talk about those 3, 2, and 1 standard deviation, what is also important is that your Levi-Jennings is almost like an expression of your distribution of your QC results. And in this chart here, in this Levi-Jennings, you see on the left side that there's actually a normal distribution which is put on its side. That is actually showing you how that distribution of these results on the right side of the Levi-Jennings is being seen. So you probably also know all the numbers that between 1 and -1 standard deviation, you will have about 65% of your QC results. Between 2 and -2, you have up to 95% of your results. So that is why if you would go wider than 3 standard deviation, between 3 and -3, you have 99.7% of all the results of a normal distribution. That shows us that outside of 3 standard deviation, we shouldn't really be expecting any more normal results. So when you look at this, for me, Levi-Jennings can really show you quickly how well you are performing. And so there is a lot more information in that Levi-Jennings that we can see. It shows us how well we've done with our settings. Is our mean and our standard deviation set okay, or is it too wide or too narrow? And is our mean also set correctly? Can we have a bias in our process or not? So when we look at the Levi-Jennings, this is one example. And so now if we look at this, do we think this Levi-Jennings looks okay? Well, if I would be looking at the distribution in here a little bit, it goes down a little bit, it goes up. There's some results between 2 and 3 standard deviation, but mainly when you look at this one, this looks like a good quality control Levi-Jennings. If we go to the next example, like this one, what do we think about this Levi-Jennings? And I would guess that most people would be, this looks very good, because all my results are nicely, tightly around the mean. But if we look at the normal distribution, what did we just say? We said that about 95% of all results should be between 2 and -2, and 65% between 1 and -1. So what do we see here is that all our QC results are between 1 and -1 standard deviation. So although this is a Levi-Jennings that most labs would be very happy with, this is actually an indication of a Levi-Jennings which has problems with the standard deviation. Your standard deviation is set pretty wide, because this distribution of the results that you see here is already a normal distribution. So what does this mean? That with this setting, you would almost need failures of 5, 6, 7 standard deviations before your Levi-Jennings or your settings here would pick up on that error. So this is actually dangerous, because this gives you a feeling that everything is well under control, but you're actually looking from too far away. You have a too wide standard deviation to really follow up the quality correctly. If we go to a next example, then this one is the opposite of what I just showed. So this also has a problem with the standard deviation, but now the standard deviation is way too narrow. So what do you see? Is you see that we have a lot of results that are outside of the 2 standard deviations and would give us warnings or rejections. This is a typical example of maybe a startup of a quality control. And what you can also see in here is because the results jump have a big jump between each other, is that this is probably something where the decimal places of the results are not detailed enough. So you also have to make sure that most of the time you can see the normal distribution. If you round results too narrow, then you might not see these. So for example, sometimes I use an A1C test, for example, NGSP, if you would say 8.1, 8.1, 8.1 every day, you'll nicely have a very stable line. But sometimes that could show you that you don't have enough decimal places and you should better start to follow up 8.14, 8.11, 8.10 to see a better variation of your data. I have a couple of other examples. If we look at this Levi Jennings, what would we think here? Well, you can see that for this one, there would be a problem with the mean. All our results are below the mean. Here you could probably also say that there might also be a standard deviation problem because my results are again very compact, very close to each other. But clearly here you have a problem. So here you would actually get a lot more rejections, false rejections probably when you have a negative or lower result. If your result starts to go up, you actually need more standard deviation, so you need to have bigger errors before you would detect a result that would be positive. And then this one is like the worst example. I hope you wouldn't see these too much, but this is clearly a problem with the mean where all my results are too low and I keep getting a lot of flags. So this could be two things. There could be a big problem with the instrument, but this could also be that just the lab had somewhere a shift, didn't adjust their settings, and is now seeing a lot of these results being way too low. So when we looked at these evaluation means NSDs and these settings, now let's take a look at crossover studies. What are we currently doing? And can we simplify our crossover based on the latest guidance of the CLSI C24 document? So what do we do most of the time in the lab? So it's very important that the lab establishes their own mean and standard deviation. And new lots of control materials should be analyzed in parallel. So it's very difficult to say, today I'm stopping, this is the last file, this is the end of my current control, and I'll start my new control now today. Those first results that you get with your new lot of control, you don't know if they're correct or not. What if there was a problem that day? You would never pick up on it if you only have one result. So what do? Well, we're using our current lot and we have the information of how our current lot is performing. We start testing our new lot. And that is what we call the crossover, the parallel testing of the new lot of material. Typically, labs have been doing this for 20 data points over 20 days. So you test it once a day together with the old lot. You don't really use your new lot yet, but you start building up your statistics. You calculate a mean, you calculate a standard deviation, and after 20 days, you use that new mean NSD and you continue with your new lot. This is what happened in most labs, and that 20 is the number that most of the labs have available. But how should we do it correctly? So the first thing we should do is, let's start with determining our new standard deviation. Well, what is the best standard deviation that we can find, or imprecision or estimate? That is the longest possible time to monitor these results. And they must include many events so that would influence that imprecision. So we would look at different operators, temperatures in the lab, reagents and reagent lot changes, lots of calibrations, lots of maintenance. All these things will make sure that our random errors, that our imprecision moves around, and that we will be able to see this. So the longer you can look at a period of time, the better you can estimate that standard deviation. So what is important is when you're looking at that standard deviation, our current lot has way more data than our new lot. So if you just run 20 points, you're only going to get a little bit of information about that standard deviation. So when we look at our current standard deviation that we have, you have to also be careful to use the correct evaluation. Remember I talked about standard deviation and coefficient of variation? Well, this is an example here where on the left side in this green bar, you can see that our current lot might be at a target of 120 with a standard deviation of 5 and a CV of 4.16%. But our new control is at 142. So let's say we would copy over our standard deviation of 5, because we say let's use the standard deviation of the old lot. That's a very good estimate. But what happened now is that because our target changed, or mean changed, we actually narrowed our imprecision. we actually went down and we're now setting a 3.52% CV. So that's not a very good way, because now we're changing our evaluation. If you look at the right side, if you have that 142, it's better to copy the CV of the current lot, that's 4.16, and then we recalculate the standard deviation to 5.9. And that will always give us the best possible evaluation by using that CV, and then either use CV all the time or bring the CV and recalculate the standard deviation. What is also important to know is that if you have short shelf life QC materials, like maybe hematology or other controls that are only valid for a couple of months, then you don't have a very long period of time to estimate a standard deviation. But what you can do is you can combine standard deviations of different lots of QCs together in this pooled standard deviation or pooled CV, coefficient of variation formula. And we would also do this if you would have like a shift in some data. Because if you have a shift like here, what happens is that if you don't use that pooled formula, you will actually increase your CV. So I put some of these numbers in here. If you see that your reagent lot A was running at a 6% CV, and then your reagent lot B was also running at 6.5% CV, but had a little bit of a different mean, what happens is if you calculate the CV over all these results, you'll actually increase your CV, and you'll see that you'll have a CV of almost 9%. So if you would say, yes?
Can you conclude in 1 minute, please?
Sure. So we're copying over our standard deviation. And so I also want to say, don't use insert values or analytical specifications for standard deviations. Don't use an interlab comparison. And then that was our standard deviation. If we look at the target mean, you can estimate a mean with 10 measurements. You don't need 20. 10 is more than enough for a mean. Try to include some calibration. Try to use the same QC frequency as your routine. And then also use, when there would be an outlier, remove that data. So same for the mean, don't use insert targets. Also, when you use multiple instruments, you can combine the data of the multiple instruments. And then as the final screen, I can show you that this would be your new crossover procedure. So what you would do is that while you're testing your current performance of your current lot, you would do 10 days and you measure 10 points. That would give you a new mean. When you have that new mean, you would use the CV of the current lot, and with that CV of the current lot, you would recalculate your new standard deviation, and now you have a new mean and a new standard deviation and CV to start running your new lot. Also remember that it can be important that after you get some more data, maybe a month in or something, you do a quick re-evaluation of your mean NSD to see if you're still well on target. And that will be it. Thank you.
Thank you very much, Nick. Thank you very much, everybody. I would like to all the panelists to switch on the camera, please. Because I was very exciting, I couldn't introduce myself in the beginning. Sorry for that. My name is Ali Ashangiti. I'm a member of Saudi Society for Clinical Chemistry and Immunology Consultant. And we will start our Q&C now. Mr. James, are you with us? Mr. James Freeman.
Sorry for that, but Dr. James and Dr. Jane, they have another meeting.
Okay.
We can connect Dr. Wim and Dr. Nico.
Okay, no problem. We can start our Q&C session. I will kick off this by one question for Dr. Helm. About the COVID antigen, do you think this will compete or replace PCR?
Okay, so this is an interesting question, whether PCR or antigen, whether antigen will replace PCR. And I think it depends on the setting. As I elaborated, PCR is very, very sensitive, which means that you may as well detect signal with PCR from a patient that is not infectious anymore. Antigen is likely to be probably better suited for large-scale demographic screening of patients than PCR. And the reason for that is that the technology is much easier to handle. It's cheaper to apply, and it's PCR and antigen both run on large automated systems. That's not the bottleneck, but I believe that that both have its place, both have its role to play. Specifically, when you, for example, would have a patient that is antigen negative but still is expressing symptoms, you may want to follow up with PCR, and PCR as well is positive in much earlier than antigen is. However, I believe that from, let's say, a mass screening perspective, antigen will probably also do the job compared to PCR. But both have its role, and they both will remain.
Okay, thank you for that. Mr. Nick, the question from the audience: in case where you have a wide established SD and your points are close to each other, however, I can't see it. However, your established SD is less than the recommended manufacturer. How can you solve this problem or this issue?
Yes, so like I said before, is that it's very important that you establish your standard deviation and your mean in your lab. So a manufacturer can give you guidance about some ranges, but you would not use that in your lab, because most of the time the ranges that a manufacturer will provide are based upon multiple instruments, could be different reagent lots and different instrument models, for example. So it is better to make sure that you always look at your own data. So that's why when you do the crossover study, you will establish those points to calculate your mean, and you will look at your standard deviation of a previous lot of control, for example, to estimate that in precision. So don't go and use manufacturer targets, but use your own targets. If your targets are wider than the manufacturer, it also doesn't make sense to stop and to reject a lot of data before. It might be important then to contact your manufacturer and ask why you have that difference, for example. But it's very important to look at your own data in your lab and to see, because if you get a better precision than what the manufacturer says, you have to protect that better precision. You have to look at that value.
Okay, Sam, what is the critical of acceptance of established means and standard deviation compared to manufacturer means? And you didn't mention the target CV from manufacturer for establishment in accepting your new establishment CV.
Yeah, so I had to go a little faster there, but for CV and the mean, both of those, I would never use the manufacturer guidance. You can look at it, and it's important to take a look if it's close enough. And often when you have a manufacturer range, your mean should be in that range. But you still always have to establish your own target, because as working with peer groups and interlab comparisons, not every instrument always gives you exactly the same result. So it is important that if your result is a little higher or a little lower than that insert value, that still you're going to monitor your instrument. It could be that your instrument has a little bit, I don't know, a higher temperature, or that the volume is a little higher. So it is important to always establish your own target and not to go solely based upon an insert value from the manufacturer. I do agree that sometimes they use that a lot for their support, or they say this is what we suggest, but you have to make sure, and all the guidance for all the documents like CLSI and other regulations always tell you to use and look at your own data and establish your own targets.
Dr. Helm, the question from my side, that's some companies produce kits using N antigen. Do you think this drawback from them side, or is it similar to S antigen?
No, So basically, there is no difference whether you want to, for antibody testing, detect the nucleocapsid or the spike protein. It makes a difference when we speak about vaccination trials because most vaccines address spike protein. So there, a quantitative spike and quantitative antibody assay makes sense. But with regards to detecting antibody response, um, N and S are equally suited to detect antibody response. However, keep in mind that we still see patients that are either N non-responders as well as S non-responders, which means that if you really want to know if somebody has antibodies and you have the suspicion that somebody may be an S or an N non-responder, you have to perform both tests to make sure that you pick up the non-responder. But it's very, very rare.
Thank you. Mr. Nick, can crossover study, what is the question? Can crossover study done in one day by running 10 points in the same day?
So in case of urgency, you could, yes, but you have to remember that if you run them all in the same day, you don't see a lot of variation because you're looking at the same calibration, the same reagent the whole time. So it is a lot better to do it over 10 days because that's why we do it over that longer period to get some more variation in there. But in an urgency, if somebody takes out the last vial of a specific lot and says there's nothing left, then you could go faster and you could repeat it a couple of times. But again, I would try to do a couple in the morning, a couple at noon, and a couple in the evening. Try to get as much variation or do it over multiple instruments if you have multiple instruments. But yes, you could speed it up in case that it's needed. But just make sure you plan it correctly and you give yourself the 10 days to do your crossover study.
Also, same question. It's interesting. If I have to, I don't know if I understood well that when I want to establish my own SD, I will use my calculated SD with the CV of the insert sheet. Second question, is there a recommended frequency for the calibration? Because I do the calibration as corrective action when the control become out.
Yeah. So I would say for the calibration frequency, that's often something the manufacturer will guide you on. Or like you said, if you are troubleshooting and you have an out of control condition, you will often use calibration to try to get the data corrected again and try to get everything well aligned again. The question in regards to using your own SD, so I always use my own standard deviation or my own CV. So again, I would not use the insert sheet CV. That is something which is there as a guidance. I would, if you start a brand new test, you have to start from scratch, then you would have to use that CV for a little bit. But as soon as you have 20, 40, 60 data points, you can establish your own CV, and then that's what you would be using to continue to evaluate your QC data.
Okay, last question for Dr. Helm. I'm sure you heard today about the approval of vaccination in UK. Do you think this will impact of the testing of antibody testing for to be mandatory for everybody? Sorry, no.
No, I was on mute. My apologies. That's difficult to say. I do believe that specifically now when we ramp up with vaccines, and this will take months, if not years, to really go large-scale vaccine campaigns, is likely that we will start with healthcare professionals and professionals that are exposed because of their job, policemen, etc. Specifically in the early days of vaccine testing, I do believe that as antigen testing, quantitative as antibody testing makes a lot of sense. However, it is also, when we look at the performance data of the vaccines that are currently being launched are very, very effective, meaning they usually have more than 90% vaccine status. At some point, it is a decision of the healthcare system. Do I have to test every individual for antibody response prior to vaccination, or given the vaccine success, do I just vaccine everybody and I don't need to do a follow-up? So I do believe that I don't think that it will become mandatory. It may be used in the beginning when we are still have to allocate vaccines to triage individuals that come with a presumed immunity. And with regards to antibody testing, I also want to draw one point, and you mentioned the S and the N discussion. It is not so important whether you look for nucleocapsid or spike. With regards to vaccination and immunity, it's important to look for neutralizing antibodies, because antibodies are not alike. You have very early immature antibodies and you have late mature antibodies that have a very high affinity and that have neutralizing effects to the virus. So if we want to discuss vaccination and immunology, then of course we should look at the correlation between the antibodies you detect and their neutralizing effects. And this can be done with, for example, with print testing. With print testing, you test live virus suppression in the presence of antibody. So more important than N and S is, of course, the capacity of the antibodies to neutralize virus infection.
Thank you very much. Thank you very much, everybody, for attending this time for this virtual booth. Have a nice day, everybody. Thanks for your time. I think this was very interesting presentation. See you, everybody. Thanks.
Thank you very much.
Thank you.
Have a nice afternoon. Bye-bye.
خلاص شباب. الو. خلاص خلص. شباب.